I know what you're thinking before we start. Oh, great. I've learned this year after year in science classes. I know how to do the scientific method, and that's great. But I thought it would be pretty helpful for us just to go through the some of the do's and don'ts of conducting a real proper scientific investigation. Because we're about to be doing them throughout the year, and we're going to do one very soon here. So it's really, I just want to touch on not to teach the scientific method to you. I know you've had that before, but more how to actually design and execute your own, because we don't want to do, for the most part this year, the standard cookbook or cookie cutter labs where someone gives you a packet or a handout and you just go through the motions and complete 10 or 12 steps and then you write a lab report. That's not a real scientific investigation. It's good and it has its place in practicing for labs and practicing some content, but it's not a real scientific investigation. So let's just really quickly, this should be a short one, go through the process step by step so you can use that to design your own investigation. We're going to use an example of something that you're probably pretty familiar with, something that you can see right over here, this guy. If you know what that is, you might know it as a roly-poly or a pill bug. They've got a bunch of different names. So let's use that as and some of its behaviors as a starting point. So first off, you know you've done scientific investigations before, but like we said, don't think of standard photocopied labs for step-by-step procedures. We want to do this as you're the scientist. You are designing the process. You decide what you're going to study. You are really doing the investigation, not just following a series of steps. You are the scientist. We want to make sure that we become really, really good at that. And it's not just following directions. It's a lot of thinking and analysis and designing. Remember, that's something I want you to be really, really good at this year. So really the process, as you remember, starts with just basic observations. We might notice some specific phenomenon that we see happening over and over again, and we really start to kind of question, well, wait a minute, and I want to know more about that. Or maybe we have some more specific questions about what we've seen and want to narrow that down to really try and learn some more. So where we had just talked about homeostasis, smiley face, briefly in class, I want to use that sort of as an example because that's what we're going to be kicking off the year with our investigations, sort of. So let's take a look at these guys. These pill bugs, roly-polies. Like we said, they've got a lot of different names, and there are many, many different species, but you've seen them before probably in nature. They're these pretty small, plated, they look like bugs or insects, but they're actually not. They're more closely related to crustaceans. Crabs and lobsters, believe it or not. But you find them underneath logs and rocks, and you've known that, and you happen to maybe notice outside repeatedly. You've been outside in mom or dad's garden or something, and you notice that, wow, I've been really seeing a lot of these. And we notice that, but we only see them under certain conditions, and that sort of puzzles you. Why do I only see them here? So for, like in this visual, this example, so let's say you notice that maybe it seems like I see them more in dark, gross places. So we got to formulate a question. The question has to be specific and it has to be testable scientifically. Now that could be in a regular lab setting. That could be just compiling statistics and data is also scientifically testing something. However you do it, you have to make sure that it is testable. So let's say our question for this is, do pill bugs prefer dark or light environments? We want to see which one is more preferable. Do they have a preference? Maybe they don't. Or we could swap out variables. Maybe they prefer saltwater versus freshwater. Maybe they prefer moist versus dry places. Again, your question is based upon shedding some light on one of your observations prior to life. So let's say we notice that, for example, instead of light or dark, let's say that you notice that they always seem to be under rocks and logs and usually in more wet, gross, slimy environments. So our question maybe could be, do pill bugs prefer wet or dry environments? So before we go any further, this is your old friend. You definitely have heard of a hypothesis before. Many of you probably were taught and learned that it was, quote, an educated guess. And that's fine, because that's sort of what it is. It's educated in that you're going on maybe some prior knowledge, some observations you've made and seen, and it's a guess in that it really should be an answer to the question that we posed. So you may sort of be guessing, like, do pill bugs prefer moist or dry environments? And what we want to do is that it's got to be testable. We have to be able to set up some kind of a test to gather data to test to see if our hypothesis is valid, and it has to actually be falsifiable. This is a big one because we really have to make sure between the two that they're testable and they're falsifiable, because sometimes we can run into trouble because a hypothesis actually can never ever be proven correct. It can only be supported by data. And luckily, if it's a sound hypothesis and it's tested over and over and over and over and over again by scientists everywhere, and they receive the same or similar results you did, then we can start to, quote, believe that hypothesis. But we want to be careful. It could never be proven correct. So really what you want to do is take the question out of the question. And we want to refrain from saying things like I think or we feel that. It doesn't seem like it's a big deal, but in true science writing, you always want to eliminate yourself from the equation. You don't want to say I or we. It's done in a passive manner. Get rid of I or we in that. And then also, don't remember from our previous video, make sure you know what independent variable is that you're testing and what your dependent variables are. Remember what you're observing. So let's go with what we had said previously. Let's say that if we wanted to test the question, do pill bugs prefer moist or dry habitats, we could actually say our hypothesis could be, notice it sounds like we pretty confident, like we know what we're talking about. Pill bugs prefer moist habitats to dry habitats. Sounds like we know what we're talking about. So that is our guess. That's our basis for what we're going to test. Now to test that, as you know, we have to have a procedure. And that is just you might have learned that as procedure, you may have learned it as materials and methods, but this is what we're going to do, the steps we're going to carry out to test our hypothesis. So we've got to gather all of our materials that we'll need, and we have to really lay this out so that specifically what we're going to do, we're testing our hypothesis. So we want to take these pill bugs and essentially, if you think about it, put them in, have the option maybe, or put them in different environments and see what happens. And it's got to be, this is very important, specific enough to be repeatable by anyone. Anybody should come in and be able to see your procedure and do exactly what you did. And if they do that and you've done a good job, they should technically probably receive very similar results to what you did. If they don't, that's going to tell us something. So like, for example, let's say we decide that we're going to set up these plastic chambers and have one side be wet, one side be dry. And when we do that, we could then say, all right, I'm going to have, notice there is an A, there's a B, and there's a C. So we're not doing it just once. We're doing it 3 different times. We want to make sure it's repeatable. And we just put a bunch of pill bugs in there, and maybe we let them decide to go back and forth through the chambers, and we just want to record data and actually see, maybe at set time intervals, how many are in the wet side, how many are in the dry side. If we put the same amounts in each side, notice, for example, 1, 2, 3, 4, 5 here, 1, 2, 3, 4, 5 here. And then record them and see what happens. Do they tend to show a preference on the wet side? Do they tend to show a preference on the dry side? So before, a lot of people miss this step, and this is key, before we actually carry our investigation, we have to carry out prediction, or make rather a prediction. So it's really testing if our hypothesis is sound, is valid, and it's supported by our experiment. What are we going to see in our results? Notice we didn't say if our hypothesis is correct. We're going to talk about later on, hypotheses can never be correct. It's always written as if-then statement. Like, for example, if, and then we would want to make sure that in the if section, that is where we would put our hypothesis when we do that. Definitely making sure that we obviously do that. And then the then in that area would be your expected results. So if, as we said, pill bugs prefer wet to dry habitats, then what should we see in our experiment? What are our results going to be that will support that hypothesis? Because if what we thought was going to happen happens, then it supports the hypothesis. If we end up with results that are completely opposite from what we thought, we've got to be clear that's not bad. That just means that, well, our hypothesis was not supported. We've got to go back to the drawing board and test out some other things. So let's say we ended up finding all the pill bugs on the dry side. That's not a bad thing. We're not quote unquote wrong. It just means that our hypothesis was not valid. We falsified it, and now we've got to go back and further investigate in another experiment to try to elaborate why were they on the dry side. So predictions are big. Remember, always written if, then. Then while we carry out our results, we need to compile our data. We're going to generate charts and graphs, making sure we record what we see. Note, qualitative and quantitative results are important. Quantitative is like over here. That's just numbers. Qualitative are qualities, things that happen, things we notice that aren't necessarily measurable by numbers, and they're equally as important depending on the lab that we do. Notice we're not in this section at this time. We're not analyzing or interpreting yet. We're not trying to figure out why this happened. We're just going over the numbers, looking at, yes, well, we can see here that it appears that a larger number over time were in the wet chamber versus the dry chamber. And you can see in this example, the person has really analyzed it mathematically. Pretty interesting. Tables, graphs, make sure everything's labeled and specific and clear. Right? And then finally, the last part is is really the most important aspect of the lab. That's where you're going to go through the results and start to analyze and interpret them. What do they tell you? What do they mean? Was your hypothesis supported or was it refuted? Notice it doesn't say right or wrong here. We really want to shy away from that. It's more, do the data support my hypothesis or do they refute it? Because even if they're refuted, that can be just as valuable. And then here's one that students often sort of forget about this one. This is a big one. Why did you obtain these results? Notice, what was the biology, the stuff that you learned in class? Like if we've just been talking about homeostasis, what does that have to do with this? Why did they prefer that? You really want to make sure that you analyze that aspect of it very, very carefully, very closely. And then we also need room for improvement. Even though we're awesome scientists, what were some weaknesses and sources of error? What could we have done differently to make our lab more scientific, more accurate? Right, so all of those are very, very, very important. So we have our basis for how to carry out an investigation. You've done it before. Now that we've gone through that, we're going to take the opportunity in class to set up our own investigations where, as we said, you're going to pick and choose what you want to do. Hope that helped. Feel free to reference the video, and I'll see you in class.